basically, and I'm going to show you how it's also organized, so all of this is going to be exactly how you would go about doing it. There's the Busczcak share folder, right, which you're all very familiar with. And you're going into there. I've put in something called apps, which I've put in some portable applications since we're not allowed to install things. I cheated and put in executable files for a few things that we could then use to make things a little bit easier. And this is also where I put my program. Then there's other people's folders, and there's your folder, right? So this is where you're going to be keeping your data. So just to quickly point out, in the apps, there's other stuff that I put in here as well. But the important things are basically that there's this XNView portable, which is what you would use to convert images if you don't save them as a JPEG or PNG or whatever. And there's Flydo, which is the thing that we're talking about. So if we go into your folder, then you're probably going to have a bunch of experiments and all kinds of data. We're concerned about this one experiment, right? So I'm just trying to make this realistic. So now this is what your data would probably look like, or something like this, before you thought about actually using something like Flydo to put it together. Basically, you're going to have a bunch of images from an experiment. You're going to have all of your stocks in some spreadsheet, right? And again, I've made sure to save this as a CSV, which is extra important. You'll find out if you didn't, because it'll just fail spectacularly. So, and then also the stocks that you're using in your current experiment, right? So you've got your master database and your subset database. So this is kind of what I figured people would have, and so I wanted to show you going from here how you actually go about creating this actual result. So just to show you really quickly, basically images, I've already organized them by some ID number, right? So here's my NID 3 and 4, 5, 6, right? And so I've already got those nicely organized. Inside of those are the actual images, and so these names don't actually matter, right? But they have to be right inside of that folder. And as you can see, these are actually TIFFs, which we'll get back to in a second. So the first thing you want to do is go back to this apps folder in Busczcak share and just copy this Flydo folder since that contains all of your programs. And then go into your folder, into your experiment, and then just put it in there. It'll take a moment, and that has all the things that I showed you previously. Remember, you also need a folder for your master databases, and it's just called master databases. And again, these names are important. If you don't name them properly, it won't be able to find stuff. And another one for your subsets. In this case, we just have one of each for simplification. Okay. So now, if we open these things up, just to show what it looks like, they're these standard CSV files. So it's just going to look something like this, right? So this is just, you got your stock names. Like I said, the ID has to be unique all down the columns. This is just an ID, 1 through 10. Has some Flybase IDs, and like I suggested, you should have a CG number or a Flybase ID in one of these columns, doesn't matter where. And this is just some random data that I made up, right? So it's got a location, it's got a source, it can have all kinds of stuff.
So does that Flybase ID refer to the stock or to the gene?
This would refer to the gene.
Okay.
Yeah, the gene reference.
So all the stocks also have Flybase ID numbers, just so people are aware.
Okay.
So that may be different.
Okay.
So the way you've organized.
Well, then actually, I guess it
it's
would give you
the
data
gene.
for that stock then.
Just for that stock.
Yeah, yeah. Okay. Yeah, was not aware of that. So that's a good point. Okay, so there's that, and again, if you were to open this in a text editor, then what you would see is, again, literally column separated values. So that's my master database, right? So I'm going to throw that in my master databases folder, and then I've got my subsets, which are the ones that I'm actually working with. So in this case, I've just chosen 4 from the original 10, right? Added some lab data, right? Got phenotypes for them, some comments about those phenotypes, right? And that's it. And again, this is a CSV file, so it's got to be this column separated there. And the most important point in all of these is that this column, the IDs, the headings can be whatever you want, doesn't matter, but the first column, the body has to contain IDs that are exactly the same as IDs from the master database. And I apologize for harping on that, but it's important. Okay, so now I've got our current experiments, and so this is the ones we're actually working with, so we can throw that in subsets. Now again, these actually could be, your subset could be your entire master database, right, just with different data associated with it, and that's completely fine. Okay, so now I've got these 2 things, and the last thing I need is images, right? And so here, I've got this images for an experiment, and like I said, inside of that experimental folder, you then have them named by ID, and inside of that are your images, and so all we have to do here is just make that base images folder, which would contain all of your experiments, right? this is just one experiment and its images, and we'll throw it in there. And that's all you need to have. The last important point is that those images are TIFFs, and so we need to convert them. So I'm going to show you how to do that. So again, it says XNView portable, and it's basically a browser just like this is, but it just happens to let you convert files into whatever you want. Okay, so we need to go to, there's a convert button over here somewhere. I believe it's this one. And then you just want to add folder and go to wherever your images folder is. It's actually, this program's pretty clever. It'll go down directories and it'll actually look for subfolders and everything and find all the images that you want. So now I'm going to go into, let me figure out where I put this. It's probably on my desktop. Okay, so this is your data, this experiment, images. And just to make sure, I'll just go to images from an experiment, right? And now you can see it found all the images. They're all labeled .tiff, and that's it. So the default settings here are probably what you want. It basically just, wherever it finds that image, it makes the new one in the same place. And it doesn't delete your old image. You can change that if you want to. You can tell it to delete the original, so that way you can delete all your TIFF files since they're really big and replace them with JPEGs or whatever you want. So basically we want our output then to be JPEG. The other important point here is, like I said, TIFF has a much higher resolution than JPEG, and so you want to make sure you go into your options and just turn up the quality as high as you can go. So put it up at best. I don't know what most of these things mean. Progressive sounds good, right? Probably. Float, best but slowest, etc. Best quality, and run it that way. And if you do it like this, you can actually see very little difference in quality. And then you hit go. This will only take a second because there are only a few files, and that's it. Now it's done. You could then go through and look at them in here if you wanted to, but it's not really necessary. Okay, so that means that we now have finished everything that you need to do. And so if we go back into your data, you would then go in, like I said, you go into your Flydo folder that has all of your scripts. And the first thing you do is build database. So you'll see a text box come up like this, and it'll think for a moment, and it'll ask you, do you want to download Bloomington and NIG stock databases? You'd only want to do this if you actually are using those databases. If you do, it'll say yes, and it'll download them to the master databases folder. It takes care of all that. You don't have to worry about it. But I'm going to say no. It also takes a while because they're pretty big files. Each one has like 12,000 lines. So we'll say no, and then it'll ask if we want to update Droid. So again, it's downloading all the Drosophila Interactive Database. Is a bunch of separate files. It takes them all separately, and it takes a while because it's something like, I don't know, 60 megabytes of files. So this is, every once in a while you might want to update it, but I don't imagine that data changes too frequently. So again, we'll say no, and then it's going to think for a minute because it's looking at the master database and the subsets and it's putting all that data together, and then it's going to go start searching the internet. And you can see it's telling you what it's doing, so that way you can see if it has an error, it'll spit out an error report and it'll say something bad happened. Mostly should work fine. So you can see it's looking at NCBI and collecting some information from there, which is stuff that's not being put on this right now, and also Flybase. And there's only 4 of these I think we did, right? So this will only take a moment. You can see it takes about this long for each one. So if you've got 200 of these, it'll take a while. And now it should be finished. There. Okay. And it should just kill itself, and that's the end. So now that we've done that, this means that if we were to go back, we would see now a summary files folder, right? And now we can see our 4 HTML files that it built, just like it was supposed to. But there's no index file. So that's the next thing you do is say, I want to make my index. You can see that took no time at all. And now we've got an index file. So if we open this guy up, then you have your completed result. So it is easier than it sounds like when I go into the details about it. But this is it. So now you can see that basically it found the experiment. So here's the images from an experiment. It told you whether or not it found images. In all these cases it did. And it's got your stock IDs that you've labeled them as. It found the gene in Flybase, and so it organized them by gene, and so on. And so then if we were to actually click into these like I was showing previously, you can actually see all the stuff that we were talking about. So it starts out with your title, right? Again, this is found from Flybase. Images, in this case, my zombie theme. So following that, we've got your actual stock source data, and then we've got lab notes. And I actually had forgotten that I have named this permanently Busczcak lab notes, and so all of yours will be called Busczcak lab notes. I can fix that if anybody really is alarmed by that or anything. Flybase data, Flybase, and so on. So this is all here, and this is the Droid results. And the Droid results are listed by database. When you actually do the search online, you'll see it all together. Here, it's going to output them separately. And that, I believe, is everything. I guess the only other thing is that if you want to say you don't like, remember I kept the TIFF so you can see they're still here. But say I don't like this picture number 5, right? This person. So I'll just delete that one. And now I want to make sure that this is reflecting. So otherwise, it's going to think it's there. It's going to look for it. And you'll see a broken image symbol. And so, what folder am I in? We'll use 3. Okay, we're going to delete number 3 here. Okay, so now if I go back into my index and have my ID number 3 and go into it, or did I delete the TIFF? Okay, anyway, the idea would be that image would be missing, right? But there would be a little box here saying there's supposed to be an image here. Most of the time you're going to be adding images, but this is just easier for the demonstration purposes. So then all you would do is go back in here and then say refresh images, just like that, and it's done. If you actually wanted to add data, in terms of stuff from your lab notes or stuff from Flybase or whatever, if you want the Flybase data to be updated, then you have to go back and run build database again, let that plod through very slowly, and then make the index. I also don't know for sure if the Flybase people get upset if somebody's accessing their website, with high frequency, which is another reason that I made sure that you don't have to do it every time, right? Most of the time they don't. I know NCPI doesn't care, but I'm not sure if Flybase is a little bit different. Yeah, so I also made a manual, which is basically exactly what I showed you, plus a few extra slides that give more detail about like what the requirements are for your databases and that kind of stuff. So yeah, this is a PDF, so you can open it anywhere. So you can see I added this, right? So, and where's this important stuff? It's mostly the same, but things like here, I just reiterating this that it must be JPEGs and so on. And then here, I give you more information about the databases and what those have to be, where they need to go. Same thing with the subset folders or files. But I think that's mostly it. And then I also added a summary, basically saying what you would do given certain circumstances, right? So this is just the overall what you do when you're first building the thing, and then this is when you want to update stuff. Like I had said, if you want to actually add new data, then you have to do basically this over again, except for all the organization. And if you just want to add images, then you just do this. And so the very end of this thing should be hopefully all you need. But I would expect that for people's specific cases, since this was designed for a specific case, and I tried to generalize it, we might run into some weird stuff that you'll have to. Feel free to ask me about. For Python to run this program to get on the network, it has to run through a proxy while it's on the school's network. The only way I could figure out how to do that was by adding a few lines to the code that are present only in the copy that's like in the Busczcak share folder here. So if you're going to run it on your machines on campus, unless you're using the guest network, then you'll want to keep that the way it is. If you want to run it at home or run it somewhere else, I can just show you what it looks like just really quickly. Let's see if I can find it. Just for example, what I've done then is added this line here. These pound signs mean comments. It's things that Python ignores. So here, proxy settings for UTSW network. Basically, you could go in here, and in this case, these are commented out because I'm on the guest network, right? So your options are basically, while you're on the network, you uncomment it, and it looks like that. When you're off the network, you can just delete them if you want to, or you can just put pound signs back in, and that's it. So that's